start recording. If someone doesn't want to appear, just please turn off the camera and the video, and will become it available for all to see later. As I was telling, this is the first edition of C3 Encontros de Ciência of this new school year. And this year, it will still be happening online through this time through a Zoom platform, and then later it will be available on our YouTube channel. I'm very pleased to thank you all for your presence and to thank Nasrin to accept the invitation to be here today presenting her work. Her work and the title of her work is How Do Live Carbon and Nitrogen Contents of Oak Hosts Affect the Heterotrophic Level of Loranthus europaeus? Insights from Stable Isotope Ecophysiology Assays. So here to present Nasrin, we have Luís Catarino, our C3 researcher, that will, and I would also like to thank Luís, he will present Nasrin in much proper way. Thank you, Luís, and thank you, Nasrin. Questions you have, you can put it through the Zoom chat, or later you can ask Nasrin directly at the end of her presentation. Thank you.
Good afternoon, everyone. Okay, today our speaker is Nasrin Seyedi. She is presently assistant professor at the Department of Forestry of Urmia University in Iran. And she is an external collaborator of the Environmental Stress and Functional Ecology group of C3. Nasrin will make a presentation about one of her latest research publications, which title is How Do Live Carbon and Nitrogen Contents of Oak Hosts Affect the Heterotrophy Level of Loranthus europaeus? Insights from Stable Isotope Ecophysiology Assays. A short note on she's career. After her graduation at the University of Kurdistan in 2003, Nasrin make a master at the University of Guilin in 2006, having a subject evaluation of quantitative and qualitative forest stands changes in Janbezara district. Nasrin did her PhD in 2011 at the Targat Modares University on Natural Resource Engineering field of forestry, with a thesis in genetic variation and architecture of Pistacia atlantica populations based on leaf morphology and progeny test. Also, the interest in the ecophysiology of Pistacia species, namely Pistacia atlantica and Pistacia lentiscus, was the base for the collaboration with C3, namely with Professor Otília Correia, here present, and Professor Cristina Mágoas, which remain up to now. Nasrin worked with us during the sabbatical period in 2010, in which she makes comparative studies on the ecophysiology of Pistacia species, which has been published as the contribution of leaf lifespan to sexual dimorphism in deciduous and evergreen Pistacia species under Mediterranean conditions, published in the Journal of Flora. After it, our collaboration with us continued up to the present. Presently, Nasrin Seyedi's main research interests are on ecophysiology of forest trees, namely carbon stable isotopes, nitrogen nitrogen stable isotope, water use efficiency, parasite plants, biotic and abiotic stresses, gender differentiation in ecophysiology of deciduous trees, and also on quantitative genetics of forest trees, progeny tests, and irritability. Nasrin Seyedi is author of many papers, most of them on forestry subjects such as species ecophysiology, population genetics, carbon and nitrogen stable isotopes, water use efficiency, parasitic plants, and biotic and abiotic stresses. Nasrin, thank you for your availability and have a good presentation.
Thank you. May I?
Yes.
Just. Good afternoon, ladies and gentlemen. It's my pleasure to have a presentation in this meeting. Today, I want to present one of my works with collaboration with Professor Ottilia Correa. How do leaf carbon and nitrogen contents of oak hosts affect the heterotrophic level of Loranthus europaeus insights from stable isotope ecophysiology assays. Loranthus europaeus is a stem hemi-parasitic plant in European forests and grows as an epiphyte on forest trees. The Loranthus genus is distributed worldwide, and Loranthus europaeus extends to the southeast and central Europe and parts of Asia. The species often develops as dense clumps in the crowns of oaks as well as of chestnut, beech, and olive species. In this picture, you can see the Loranthus on top of the oak trees. Mistletoe are parasite angiosperm plant that attack the xylem of their host through a specialized organ, the hostorium. Here you can see the hostorium that can attack the stem. They are stem parasitic and can only access dynamic organic compounds present in the host's vessel elements, such as organic nitrogen and organic carbon, most probably available in the form of amino acids. Here you can see the hostorium can attack the stem and then germinate. And after that, they affected the growth of trees. Parasitic plants alter the morphology and physiology of their hosts and lead to an abnormal growth pattern by diversion of host resources. The net effect of these changes on physiology and morphology is that growth rates of infected trees may be reduced by as much as 80%. In these pictures, you can see the abnormal growth of infected trees. Parasites by the mistletoe affect the water, nitrogen, and carbon cycles of the host plants, both at an individual and at a larger scale, especially in arid and semi-arid regions, where water and nutrient limit ecosystem dynamics. Next to water, available nitrogen is the most important limiting factor in the production of dry land ecosystems. The analysis of stable isotope ratios has an important role in providing information on the dynamics of carbon and water and nutrients in parasitic plants and their hosts. For instance, the proportion of host-derived carbon in the hemi-parasite biomass can be calculated using the stable isotope of carbon. The high stomatal conductance and transpiration rates of parasites drive rapid import of solutes via the xylem. So, carbon intercellular concentration is relatively high and the carbon isotope fractionations is stronger in mistletoes, which causes carbon-13 composition to decrease more in the hemi-parasites than in their hosts. Therefore, high transpiration and low carbon assimilation under conditions of limited nutrient supply result in a decreased water use efficiency in a mistletoe compared with of its host, and it can be detected by a difference in carbon isotope composition. Previous studies showed that the differences in delta-13C values between mistletoes and host decrease with increasing nitrogen content of the host tissue. Based on this result, we also examined the effect of carbon content of the hosts on the differences in delta-13C values between them and the mistletoes. The consequences of this effect on heterotrophy have not been reported so far. On the other hand, one of the main leaf variables is leaf mass per area, which is highly correlated with various functional leaf traits. Previous studies reported various relationships between LMA and leaf nitrogen concentrations. The hypothesis is that with increasing leaf carbon content and LMA of the host mistletoe carbon 13 composition difference was decreased and the degree of heterotrophy of mistletoe increased. The objective is that to what extent do the leaf carbon and nitrogen contents and LMA of the host affect the heterotrophic degree of the mistletoe? We did this study in Iran. The sample is Iran here. You can see the location of Iran and in compare with the Portugal. Iran is a big country with various climate. And right now I'm in Iran and we sampled from this part in the northwest of the Iran. Zagros forest is the semi-Mediterranean climate that is classified as humid. Selected species were Quercus libani, Quercus infectoria, and Quercus brantii. 10 mistletoe host pairs of each species were sampled and leaves were collected from them. And the parameters that we measured in this study were LMA and also leaf carbon and nitrogen contents on dry mass basis and a stable isotope composition that we did on Faculty of Science, University of Lisbon. You know better than me. And the heterotrophy of the mistletoe was calculated from these equations. Some statistical analyses were done, for example, colomograph smear node test for ensuring the normal distribution, pair t-test to compare the studied parameters between mistletoe and host, and annual and Duncan average test to find the differences among the species. And then we did also Pearson correlation in SPSS, and after that we did SMA analysis for significant relationships in R softwares. Results. If I want to report results, it's better to be in two sections. First part is based on ANOVA, and on that we compared the differences between the species in hosts and mistletoes on different hosts. Based on ANOVA analysis, Quercus infectoria has the highest value of LMA from the host and also from the mistletoe, and the significant differences between the species in each columns show in different letters and based on ANOVA. So Quercus infectoria is different in LMA with Quercus libani and Quercus brantii, and also in carbon content, nitrogen content, all of them, Quercus infectoria is the has the highest value, and it has a significant difference between species. But in the carbon isotope composition, we didn't see any significant differences between species, and also in nitrogen 15 composition. In the second part, we compare between host and mistletoe in pair t-test, and I showed a significant difference in numbers, and you see for all the parameters there was a significant difference between host and mistletoe in Quercus libani, in Quercus infectoria, Quercus brantii, and for LMA for carbon concentration, and for nitrogen concentration, and also for delta 13C, we observed a significant difference between host and mistletoe. But in delta 15N, we didn't find any significant difference between host and mistletoe. Table 2 is the ANOVA of this analysis that I described in table 1, and here we have a t-test that I described in table 1, but here you have a detail in each species and difference between host and parasite that I described in table 1. Based on Pearson's correlation and SMA analysis, the correlation between delta 15N of host and mistletoe was significant in Quercus libani and Quercus infectoria. Here you can see the Pearson correlation and SMA in our software. Based on the scatter plots and linear regressions, the highest value of regression coefficients was observed in Quercus infectoria. Here in Quercus libani, you see the regression coefficients. In Quercus infectoria is more than that, it's 0.80, and then in Quercus brantii. For the heterotrophic degree, we found that heterotrophic degree of mistletoe on Quercus infectoria is the highest value. It's around 60%, and in the Quercus libani is the minimum value is 23%. So the highest value of heterotrophic degree is the mistletoe on Quercus infectoria. Discussion. In this study, there was a significant correlation of delta 15N between mistletoe and host, but with similar values of these parameters in both. This indicates that there is no isotope discrimination during nitrogen absorption by the mistletoe, and confirms that it receives all its nitrogen from the host. A strong correlation between delta 15N of host and mistletoe has also been reported in other studies. Because soil nitrogen is a non-limiting factor in Zagros forests, the host plants are not expected to fully meet the parasite's demand for nitrogen. Therefore, host nitrogen can play a decisive role in the rate of parasite heterotrophy, as in general, parasites prefer trees with high nitrogen content. In our study, the mistletoe had a lower delta 13C than the host in all 3 oak species. It can be stated that the studied hemi-parasite has a lower water use efficiency than its host. The host also had a higher nitrogen use efficiency than the mistletoe. The positive relationship between water use efficiency and nitrogen use efficiency is possibly due to the fact that when using more water, the plant increases its photosynthetic capacity and has higher stomatal conductance than when it uses less water, leading to higher nitrogen uptake. In this study, the mean LAMA of Quercus infectoria was higher than that of Quercus libani and Quercus brantii. The lowest value of carbon-13 composition was observed in Quercus infectoria, and this means that Quercus infectoria trees have low water use efficiency as well as low nitrogen use efficiency, and that their mistletoes will extract more nitrogen. In this study, leaf carbon and nitrogen contents differed significantly among the 3 oak species, and the highest values were those of Quercus infectoria. On the other hand, mistletoe of Quercus infectoria had the highest amount of leaf carbon as well. This suggests that organic nitrogen compounds from the host xylem are providing carbon as well as nitrogen to the mistletoe. The relative degree of heterotrophy rises with the nitrogen content of the host, probably due to additional sources of organic material other than amino acids. The supply does not need to be continuous. For instance, it may be a flush of carbohydrates at the time of bud burst, early in the wet season. Thus, we can hypothesize that the assimilation of a larger amount of amino acids and other compounds relatively enriched in carbon-13 and dissolved in the host's transpiration stream caused the delta 13C of mistletoe to be similar to its host and thereby reduce the difference between them. Previous studies reported that the difference in delta 13C between host and mistletoe was least in the case of hosts with high nitrogen contents. However, most of studies were in arid or semi-arid regions and suggested that this may not be true in humid areas. In this study, a logical relationship was found between carbon and nitrogen with host mistletoe, carbon-13 composition difference, as well as with the degree of heterotrophy. Also, there was a logical relationship between host LAMA and host mistletoe delta 13C difference, as well as with heterotrophy. According to our initial hypothesis, the highest level of LAMA, carbon, and nitrogen concentration were observed in Quercus infectoria with the highest degree of heterotrophy and the minimum differences in carbon-13 composition between host and mistletoe. The heterotrophic degree of mistletoe was positively related with oak leaf mass per area and with carbon and nitrogen contents on a mass basis. This suggested that in humid regions, host LAMA, carbon, and nitrogen content can affect the degree of mistletoe heterotrophy, and host mistletoe delta 13C difference is least on hosts with high LAMA, carbon, and nitrogen content. Since the highest mistletoe LAMA was also observed in Quercus infectoria, our results showed that leaf mass per area of both host and mistletoe can be indicators of the heterotrophic degree of mistletoe and can be used instead of delta 13C as a low-cost and easily measurable alternative. It's hoped that the current study will simulate more detailed studies emphasizing the importance of incorporating mistletoe associations in budgets of carbon, water, and nutrient in humid regions. Thank you for your attention.
Thank you very much, Nasrin. So we have plenty of time to questions. I don't know who wants to start. Yes.
If someone have questions, I can reserve my one to after. But thank you, Nasrin, for your interesting presentation. Indeed, the topic of mistletoes is also very interesting to me. I am working in on a research and development project about the enemies of cashew tree, Anacardium occidentale, in Guinea-Bissau. It is also an Acardiaceae species, like the pistachias. But this is in Guinea-Bissau. And one of the biggest problems in some orchards is the infestation by species of Loranthus, namely from the genera Agelanthus and Tapinanthus. They are probably propagated by birds that eat the fruits and release the seeds in the faces on the host trees. I think these Loranthus species are not very selective in terms of host species, because they are using Anacardium occidentale, but they are also present in the forests, in the savannas, in several tree species from local flora. From what we know, do you know also that these parasitic species are not selective in terms of hosts? And do you know, or can you suggest any control measures to deal with this problem? It's my that are my questions.
In our forests also, Loranthus is a big problem for our forests also. And some of my colleagues, they study about how can we control of the Loranthus. Actually, I didn't work about how can we control of the Loranthus, but I can ask them, and of course I will email you what they did for the control of these parasites in our forests.
Okay, thank you.
You're welcome.
So I don't know, any other question? Any? No? Give 1 minute, just if you don't have questions. Well, the presentation, and Nasrin is here with us, but the presentation will be available, and if you want then to contact Nasrin, I think it's okay. Yes, Nasrin?
I didn't put all of the parts in the presentation because it's will be very take very time, but the paper is available in ecological indicators, and I will be available if everyone has a questions.
Okay.
And so on.
So yes, so I think we are going to finish our talk here and the recording also. Thank you again once more, Nasrin, for your presentation and to be here with us this first Encontro Ciência of the year. Next week we will have another one, so I invite you all to be present in next Thursday, and I will put the video on the Heterotrophic YouTube channel in very soon. So thank you all.
Can I thank you. I thank you for inviting me for the meeting, and special thanks to Professor Ottilia because her collaboration and her helps.
Thank you.
Thank you.
So see you next week. Bye. Thank you all.
Bye. Thank you.
Bye bye.
Thank you. Bye bye.